Hello learners, in our today's class we have to start with another pathway of antigen presentation, that is exogenous pathway of antigen presentation. The exogenous pathway of antigen presentation is also referred to as the endocytic pathway of antigen presentation. It is referred to as endocytic pathway of antigen presentation also because the organelles which are involved here include endosomes and lysosomes. One most important thing that you need to know about antigen presentation here is that the antigen which has to be presented through the MHC, and that also specifically through the MHC2, has to come from outside. In comparison, when we talked about the endogenous pathway, there the antigen had to be synthesized within the cell itself. The organelles which are involved here include endosomes and then the lysosomes. If we look into the molecular structures and the details of the cell, we come to know that this is the antigen which is coming from outside into the cell. Now inside the cell it can enter through two ways. One is phagocytosis, the second is endocytosis. When we talk about phagocytosis and the endocytosis, in both of the conditions it has to be engulfed inside the cell and then it has to be encapsulated within the cell. Now let us see those cells which present or which perform phagocytosis and those cells which perform endocytosis. So if we talk of the antigen presenting cells, which are the APCs, so the APCs only are the ones which will present MHC2. Out of the APCs, the macrophages are the ones which will perform phagocytosis. But the other cells, including dendritic cells as well as including the B lymphocytes, will perform endocytosis. Since majority of the cells are performing endocytosis, we call it as endocytic pathway. Now as I was continuing, the antigen enters inside the cell and it is enclosed in a vesicle. This vesicle is generally coated with the proteins called clathrins. So these are called as clathrin coated vesicles. These clathrin coated vesicles have to then either be fused or either be converted into early endosome. The early endosome has the pH of around 6 to 6.5, means it is very slightly acidic. But as this further transits into the late endosome and then the lysosome, the acidic conditions will increase. Means the main purpose of endosomes increasing acidic conditions is that the antigenic peptide has to be degraded into the smaller peptides. Since here the proteasomes are not involved, then how can we degrade the antigen into the smaller peptides? For that, the increasing acidic conditions will suffice the work of proteasome, because as the acidic conditions increase, the peptides cannot be stabilized and hence they denature as well as they break down into smaller pieces. So the antigen's early endosome will now transit into the late endosome and then finally into the lysosome. During this time the pH will be decreasing continuously so that the antigen degrades into smaller peptides. In the meantime, what happens is that the MHC2 here will be coming from the RER, will be packaged into the Golgi, and finally it will be fusing with the endosome itself, and there it will combine with the antigen. Unlike MHC1, where the antigen had to first enter the rough endoplasmic reticulum. Now let us look into this part wherein how the antigen is presented in the rough endoplasmic reticulum and how then it is going into the how the MHC2 is presented in the rough endoplasmic reticulum and then how it is going to the Golgi complex. Now when the MHC2 molecule is synthesized in the rough endoplasmic reticulum, there is one very important molecule which remains bound to this MHC2 molecule throughout the process. This molecule is referred to as invariant chain. The invariant chain will continuously remain bound to the MHC both chains, that is alpha and beta. This invariant chain is very important and it has the cluster of differentiation 74. You can say that there can be almost and up to maximum 3 pairs of alpha and beta chain which can together bind to a single invariant chain at a time. Invariant chain itself is a trimeric protein and it will bind and interact with the peptide binding cleft of the class 2 molecules. Now what is the main importance of binding of this MHC with this invariant chain? Because once it is inside the rough endoplasmic reticulum, the same way there also the MHC1 complexes are also present. Now for MHC1, the antigen comes inside the rough endoplasmic reticulum, but for MHC2, the antigen is present outside. In order that the antigens for the MHC class 1 do not bind to the MHC class 2, the invariant chain will mask the peptide binding cleft so that these green colored antigens do not bind to the wrong molecule. So this is the main function of invariant chain. That is, it will block the binding of endogenous antigens. Now we will talk about that. How will it be removed? Till the time it is in the rough Now as it moves out of the rough endoplasmic reticulum, then it eventually degrades into the smaller peptides. Why this happens? Because once it is moving out of the rough endoplasmic reticulum, it has to eventually merge into the endosome. Now it is noteworthy that endosome has less pH, means it has more acidic conditions. In that case, the invariant chain, because it is being a peptide, it has to be or it will be degraded. So what happens is that after it moves out of the rough endoplasmic reticulum and then it has been packaged from the Golgi, and when it enters the endosome, only a part of it remains, that is clip. The remaining of it is degraded. The two main functions of the CD74 or the invariant chain was that firstly it will not allow the class 1 antigens, which we also call as the endogenous antigens, to bind to the MHC2. Secondly, it will also allow the proper folding of MHC2. We already know this, that inside the rough endoplasmic reticulum, if I need to draw the diagram to explain this, if this is the rough endoplasmic reticulum, we already know that here there is MHC class 1 molecule also being processed, and here there is MHC class 2 molecule also being processed. Now the antigen has to bind, which is endogenous in nature, has to bind to the MHC class 1 molecule. In case if it goes on and if it binds to this part, then it will be a problem. So in order to avoid that, the invariant chain plays a role because it will be masking the peptide cleft of the MHC class 2. After this, as I already told you, now this will be packaged into the Golgi and later on it has to be traveling into the endosome. In the endosome, because the pH is less, the invariant chain will be degraded and only a part of it will be left, that is clip. Now there is this exogenous antigen. This exogenous antigen also in the endosome will be fragmented into smaller peptides because the endosomes have very low pH. One of the peptides of it now has to attach to the cleft of MHC class 2. Now this will be mediated by HLA-DM. Now what is HLA-DM? It is a molecule which will help to bind this particular antigenic peptide into this cleft, and also to remove this clip, the HLA-DM will play its role. The HLA-DM can be clearly observed here. This is the class 2 MHC molecule and this is the invariant chain. The invariant chain was bound to this until the HLA-DM molecule comes. The HLA-DM molecule is basically trying to remove the clip and help the peptide to attach to the cleft of MHC2. So this is a very important thing which you need to know. And this HLA-DM is a non-classical MHC2. Like we talked about the non-classical MHC1 also, this is non-classical MHC2. Later on, finally, when the antigenic peptide has bound to this MHC class 2, now it will be presented through the APC, which is here. For the example, let's say it is the B cell or the B lymphocyte. Now here the CD4 plus cell is present, and its TCR will be recognizing this particular MHC class 2, and then it will be functioning accordingly. So in this way the exogenous pathway of the antigen presentation works. Now let us try to understand the endogenous pathway of the antigen presentation as well. Means we are going to compare it in the comparative format. So here was the endogenous antigen. We are firstly talking about the endogenous pathway. So the endogenous antigen is that one which is synthesized within the cell. It is the rough endoplasmic reticulum where it has to be transported. So now this endogenous antigen will first be processed into the proteasome, wherein it will be degraded into smaller peptides. These smaller peptides through TAP will be entering into the RER. In the RER, the MHC1 is already being processed with the help of calnexin, calreticulin, ERP57 as well as stepacin. This mature MHC1 will now bind to the antigen. In order that this antigen doesn't bind here in this wrong place, the invariant chain is continuously masking the class 2 MHC molecule when it is in the rough endoplasmic reticulum. Now both of them will be traveling outside, be it MHC1 or be it MHC2. Noteworthy point is, when they are in the Golgi complex, the MHC1 is already bound to the antigen, but the MHC2 is not bound to the antigen yet, it is bound to the invariant chain. Now here the function of this whole MHC1 is over, and the processing of the MHC1 is over. It has to now directly go and represent itself out of the cell. But the MHC2 functioning now will mainly start. It will first enter the endosome, wherein because of the acidic conditions only the clip part will remain. Later on the antigenic peptides will be attached here with the help of the HLA-DM, and the clip part will also be removed with the help of HLA-DM. Then finally this MHC2 will be bound to the antigen, and this molecule will be presented out of the cell. And then finally we know that this class 1 MHC molecule will be recognized by CD8+, but the class 2 MHC molecule will be recognized by CD4+, and then accordingly the functions will start. So this is the comparative account of class 1 and class 2 MHC molecules.